Hey everyone, today we are going to learn regulation of glycolysis, and I first want to quickly review the steps of glycolysis. So in the first step, where glucose is converted into glucose 6-phosphate and the reaction is catalyzed by enzyme glucokinase and hexokinase. And there are three important things to remember about this reaction. A. This is the first irreversible reaction of glycolysis. B. This step ensures that glucose is committed further into the glycolysis pathway by trapping glucose into glucose 6-phosphate inside the cells, since glucose 6-phosphate cannot cross the plasma membrane to transport it back into the extracellular space. And third, this is the energy consuming reaction which requires ATP to convert glucose into glucose 6-phosphate. Now, glucose 6-phosphate is then subsequently isomerized into fructose 6-phosphate, and then fructose 6-phosphate is then converted into fructose 1,6-bisphosphate, and the reaction is catalyzed by phosphofructokinase 1 enzyme. And this is the second irreversible step of glycolysis pathway, which also requires ATP to convert fructose 6-phosphate into fructose 1,6-bisphosphate. So this is the first phase of glycolysis, which is also called as energy investment phase. Now, fructose 1,6-bisphosphate is then subsequently cleaved into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And because only glyceraldehyde 3-phosphate can further enter into the next phase of glycolysis pathway, dihydroxyacetone phosphate must isomerized into glyceraldehyde 3-phosphate. Therefore, from one glucose molecule, now we have two molecules of glyceraldehyde 3-phosphate. Now, subsequently, glyceraldehyde 3-phosphate is oxidized into 1,3-bisphosphoglycerate, and this is the reaction where NAD plus is reduced to NADH, and 1,3-bisphosphoglycerate is then converted into 3-phosphoglycerate, and this is the reaction which also synthesizes two molecules of ATP, and 3-phosphoglycerate is then converted into 2-phosphoglycerate, and dehydration of 2-phosphoglycerate synthesizes phosphoenol pyruvate, which is then subsequently converted into pyruvate with the help of enzyme Pyruvate Kinase, which synthesizes two molecules of ATP. And this is the third irreversible reaction of glycolysis pathway. And lastly, pyruvate is reduced into lactate with the help of enzyme lactate dehydrogenase, and in this reaction NADH is oxidized to form NAD plus. So now you can see that the NAD plus which was lost in glyceraldehyde 3-phosphate reaction, it is now reobtained when pyruvate is reduced to lactate. So overall, there are three important irreversible reactions of glycolysis pathway, and these reactions are carried out by enzyme hexokinase, phosphofructokinase 1 and pyruvate kinase, and therefore these enzymes are under tight regulation to control the rate of glycolysis. And before we discuss about the regulation of this enzyme, how they regulate glycolysis pathway, I first want to quickly discuss the two important mechanism of enzyme regulation. So the first mechanism of enzyme regulation is called allosteric regulation. In allosteric regulation, the enzyme has two binding pockets. So the first one is called active site. This is the site where substrate binds to enzyme, and the other site is called allosteric site. In allosteric site, regulatory molecules, which could be either activator or inhibitor, binds to an enzyme, and this is the site which must be different than the active site, and therefore it is called allosteric site. Now, allosteric activation or inhibition of an enzyme is short term, which usually lasts from minutes to hours. So this is the allosteric regulation. Now, the next enzymatic regulation is performed by hormones. So unlike allosteric regulation, hormones influences the amount of enzymes being synthesized, and it regulates the amount of enzymes being synthesized by modulating gene transcription of the enzyme. And most importantly, Depending on the nutrient condition of the cell, hormones mediated modulation of enzymatic activity is long term, which could last from hours to days. So now we know the two important mechanisms of enzymatic regulation, that is allosteric regulation and hormonal regulation. Let's talk about how the first reaction of glycolysis is regulated. So in the first reaction of glycolysis, both hexokinase and glucokinase enzyme catalyzes the entry of glucose into the glycolysis pathway by phosphorylating glucose into glucose 6-phosphate. And remember that both these enzymes, they are different, however they catalyze the same reaction, that is converting glucose into glucose 6-phosphate. Therefore these two enzymes are considered as isozymes. They are nothing but they are different proteins that catalyze the same reaction. And humans have 4 isozymes of hexokinase enzyme, that is hexokinase 1, 2, 4. First is hexokinase 1, 2, 3. So isoform 1 is present in brain and red blood cells. Isoform 1 and 2, they are present in muscle, and all these isoforms are also present in other metabolizing cells and tissues except for liver and pancreas. Whereas hexokinase 4, which is also called as hexokinase D and also called as glucokinase, which is predominantly present in liver and pancreas. So I will be referring this hexokinase 4 enzyme as a glucokinase so that we don't get confused between hexokinase and glucokinase. When I say hexokinase, it involves all 3 isoforms 1, 2 and 3, whereas when I say glucokinase, it means that is hexokinase 4. So now let's talk about the regulation of hexokinase enzyme. So hexokinase enzyme is allosterically activated by its substrate glucose. So when glucose is entering into the cell, it activates hexokinase enzyme, and hexokinase then initiate the conversion of glucose to glucose 6-phosphate. On the other hand, hexokinase is allosterically inhibited by its product glucose 6-phosphate. So when cellular concentration of glucose 6-phosphate rises above its normal level, it temporarily and reversibly inhibit hexokinase enzyme until the glucose 6-phosphate levels brought down to normal via its consumption through glycolytic pathway. So until the glucose 6-phosphate levels are not normalized, it can allosterically inhibit hexokinase enzyme, and once it is consumed and bring back to the normal level, it will then release its inhibition on hexokinase enzyme. So overall, the hexokinase enzyme is allosterically activated by its substrate glucose, whereas it is allosterically inhibited by its product glucose 6-phosphate. So now let's talk about how glucokinase enzyme is regulated in the liver. So unlike hexokinase, glucokinase enzyme is not allosterically inhibited by its product glucose 6-phosphate. Instead, glucose 6-phosphate is allosterically inhibited by fructose 6-phosphate in the liver. And there is one very interesting mechanism how fructose 6-phosphate inhibits glucose 6-phosphate in the liver. So in the presence of low blood glucose, fructose 6-phosphate inhibit glucokinase by translocating glucokinase in the nucleus. And in the nucleus, glucokinase binds with nuclear protein, which is called glucokinase regulatory protein. So glucokinase regulatory protein actually indirectly inhibit glucokinase by trapping glucokinase into the nucleus and renders it inactive, and therefore glucokinase cannot involved in converting glucose into glucose 6-phosphate, and hence the glycolysis pathway is inhibited. On the other hand, in the presence of high blood glucose, for instance after having carbohydrate rich diet, glucose enters into the cell through GLUT transporter. As a result, glucose concentration in the cytosol increases, and the higher glucose concentration in the cytosol equilibrates with glucose in the nucleus by transporting through the nuclear pores. And glucose inside the nucleus causes the dissociation of glucokinase and dissociation of glucokinase from glucokinase regulatory protein and enables the glucokinase to re-enter into the cytosol and begins phosphorylating glucose into glucose 6-phosphate. This means that similar to hexokinase, glucokinase is also allosterically activated by its substrate glucose. Now let's talk about how hormones regulate glucokinase enzyme in the liver. So when blood glucose levels are high, there is an increase in secretion of insulin, and insulin activates glucokinase enzyme by inducing its synthesis through increasing the gene transcription. On the other hand, when blood glucose levels are low, that leads to increase in glucagon levels, and glucagon inhibits glucokinase enzyme by inhibiting gene transcription, therefore it inhibiting the synthesis of glucokinase enzyme in the liver. Now the main purpose of slowing down glycolysis pathway in the liver is to stop glucose consumption by liver and spare glucose for other organs, and most importantly for brain, because brain solely rely on glucose to synthesize ATP unlike other organs. And in the presence of high blood glucose, when there is too much of glycolysis pathway going on, it also produces too much of ATP. So in that case, when there is too much of ATP is being produced, then it is being utilized, then glucose 6-phosphate can divert into glycogen synthesis pathway in the liver. That means glucose can be stored as glycogen in the liver, and this glycogen then can be broken down into glucose when there is a need of energy. So depending on the nutrient status of the cell, glycogen can be broken down into glucose, and then glucose can be used to synthesize ATP. So this is the allosteric and enzymatic regulation of hexokinase and glucokinase enzyme, which is involved in the first irreversible reaction of glycolysis pathway. So now let's talk about the second irreversible reaction of glycolysis. So the second irreversible reaction of glycolysis, which involves the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate, and the reaction is catalyzed by phosphofructokinase 1 enzyme. So pyruvate is the end product of glycolysis, and it is then subsequently metabolized in the TCA cycle in mitochondria to synthesize ATP. However, if cell synthesizes too much of ATP, it indicates that ATP is being produced faster than it is being consumed. And this causes the accumulation of ATP inside the cell. As a result, there is an increase in the ratio of ATP to ADP. So when ATP is being consumed, it is broken down into ADP and AMP. And therefore, when the ratio is increased, it indicates that the ATP production is higher than its consumption. And furthermore, the increase in the ratio also indicates that there is an abundant energy supply within the cell. That means these cells are called energy-rich cells. And therefore, there is a need to stop producing more ATP inside the cell. So the accumulated ATP itself can allosterically inhibit PFK1 enzyme. So ATP actually binds to the allosteric side of the enzyme, and as a result, the enzyme, there is a reduction in the affinity of the enzyme for its substrate fructose 6-phosphate, and therefore the rate of glycolysis is reduced in the cell. Now consider the opposite situation where consumption of ATP is higher than it is being produced. This will result in the decrease in the ratio of ATP to ADP. So high ADP level indicates that there is an increase in the ATP consumption, and this causes the accumulation of ADP and AMP in the cells. And the decrease in the ratio also indicates that the cell's energy stores are depleted. So therefore, these cells are called energy deficient cell, and therefore these cells need more ATP production, and to produce more ATP, this accumulated ADP and AMP allosterically activate phosphofructokinase 1 enzyme, and by activating PFK1, it also relieves the inhibition on PFK1 by ATP, and therefore it increases the rate of glycolysis in the cell. Now, when cells are producing too much ATP, it not only accumulates ATP in the cells, but it also saturates TCA cycle, and as a result there is an increase in the level of TCA cycle intermediate, which is called citrate. So citrate translocate from mitochondria into cytosol, and the citrate level, and there is an accumulation of citrate in the cytosol. Now citrate can be formed by oxidation of either pyruvate fatty acids or amino acids. An increase in the cytosolic citrate level serves as an intracellular signal which indicates that cells are meeting its current needs of energy from oxidizing fats and amino acid. That means it is indirectly saying, the accumulation of citrate indirectly saying that it is already getting enough energy supply from oxidizing fats and proteins, so it does not need glycolysis pathway to synthesize more ATP. Therefore, citrate allosterically inhibits PFK1 enzyme to slow down the rate of glycolysis reaction in the cell. Furthermore, citrate not only inhibits the glycolysis pathway, but it actually favors the use of glucose for glycogen synthesis. In that way, glucose 6-phosphate is diverted for glycogen synthesis pathway. So glucose is being stored as a glycogen, and depending on the nutrient status of the cell, glycogen can later be broken down into glucose to synthesize ATP. Now, in addition to ADP and AMP, there is another molecule which allosterically activate PFK1 enzyme, and this is called fructose 2,6-bisphosphate. In fact, fructose 2,6-bisphosphate is a potent allosteric activator of PFK1 enzyme, and because it is a potent allosteric activator, the level of fructose 2,6-bisphosphate is tightly regulated in the cells. So let's talk about fructose 2,6-bisphosphate. So here we know that fructose 6-phosphate is converted into fructose 1,6-bisphosphate with the help of enzyme PFK1. Now, this small amount of fructose 6-phosphate can also be converted into fructose 2,6-bisphosphate with the help of enzyme PFK2. On the other hand, fructose 2,6-bisphosphate can be broken down into fructose 6-phosphate with the help of enzyme fructose bisphosphatase 2, that is FBP2. So remember that this is the kinase, means kinase actually adds the phosphate group onto the molecule. That means it phosphorylates the molecule, whereas phosphatase enzyme, the main function of phosphatase enzyme is to remove the phosphate molecule from the molecule. That means it dephosphorylates the molecule. So kinase is phosphorylates and phosphatase is dephosphorylates the molecule. So the interesting thing about PFK2 and FBP2 are, in actuality, they are just one single protein which has two separate enzymatic activities. This is one single protein which has two separate enzymatic activity. That means PFK1 has a kinase activity which phosphorylates the molecule, and FBP2 has phosphatase activity which dephosphorylates the molecule. And most importantly, the cellular level of fructose 2,6-bisphosphate is regulated by insulin and glucagon in liver. So let's talk about how this fructose 2,6-bisphosphate levels are regulated in the liver. So in the presence of low blood glucose, the glucagon level is high, and higher glucagon level stimulate the adenyl cyclase enzyme, which then increases the synthesis of cyclic AMP, and cyclic AMP, increase in the cyclic AMP then further activates protein kinase A enzyme. So protein kinase A phosphorylate the PFK2 and FBP2 enzyme, which enhances its phosphatase activity. So when phosphatase activity is enhanced, it breaks down fructose 2,6-bisphosphate into fructose 6-phosphate. So to summarize, increase in glucagon level in response to low blood glucose decreases the level of fructose 2,6-bisphosphate, which result in the inhibition of phosphofructokinase 1 enzyme as well as inhibition of glycolysis pathway. On the other, at the same time, a decrease in the fructose 2,6 bisphosphate level also activates the gluconeogenesis pathway in the liver to synthesize more glucose. And this increase in the production of glucose also enables the liver to replenish blood glucose in response to higher glucagon level. So, together, glucagon lowers the cellular fructose 2,6 bisphosphate level, inhibiting glycolysis and stimulating gluconeogenesis in the liver. Now let's talk about in the presence of high blood glucose. So when blood glucose levels are high, it results in the increase in the high insulin secretion, and high insulin secretion activates protein phosphatase enzyme, and protein phosphatase enzyme catalyze the removal of phosphate group on the enzyme, and therefore enhances the kinase activity of this enzyme. So when kinase activity is enhanced, it converts fructose 6 phosphate into fructose 2,6 bisphosphate. Therefore, when insulin levels are high in the presence of high blood glucose, it increases fructose 2,6 bisphosphate, and fructose 2,6 bisphosphate, since this is the potent allosteric activator of PFK1 enzyme, it activates PFK1 enzyme, increases the rate of glycolysis. At the same time, it also inhibits the gluconeogenesis in the liver. So higher insulin level in the presence of high blood glucose increases fructose 2,6 bisphosphate, increases rate of glycolysis, whereas inhibiting the rate of gluconeogenesis in the liver. So main function of fructose 2,6 bisphosphate is to make sure that both glycolysis and gluconeogenesis are not activated or inhibited at the same time. It is to make sure that these two pathways are regulated according to the need of the cells, or according to the nutrient status of the cells. So this is the overview of allosteric and hormonal regulation of phosphofructokinase 1 enzyme, which carries the second irreversible reaction in the glycolysis pathway. So now let's talk about the third enzyme, that is pyruvate kinase, which catalyzes the third irreversible reaction in glycolysis pathway, and this involves the conversion of phosphoenol pyruvate to pyruvate, and PEP is basically abbreviated as phosphoenol pyruvate. So pyruvate kinase is allosterically inhibited by ATP, acetyl-CoA, and long chain fatty acid. All these three molecules are indicative of abundance of energy within the cell. So when these molecules are activated in the presence of energy, it allosterically inhibit pyruvate kinase. So basically it binds to the allosteric side of the pyruvate kinase enzyme, and therefore it reduces the affinity of pyruvate kinase for its substrate phosphoenol pyruvate, and therefore reducing or inhibiting the rate of glycolysis. On the other hand, pyruvate kinase enzyme is activated by fructose 1,6 bisphosphate, and fructose 1,6 bisphosphate is the product of phosphofructose kinase 1 enzyme. So when pyruvate kinase enzyme is activated by fructose 1,6 bisphosphate, this is an example of feed-forward reaction, and feed-forward reaction is to make sure that the reaction is moving in the forward direction. To say it differently, fructose 1,6 bisphosphate kind of giving a heads up to pyruvate kinase enzyme for the increased amount of workload coming ahead on its way. So it's almost preparing pyruvate kinase enzyme for the amount of workload it has to carry to ensure that the glycolysis pathway is moving forward. Now, pyruvate can also be converted into alanine through transamination process. So when there is too much of alanine is being produced, the accumulation of alanine can also allosterically inhibit pyruvate kinase enzyme. So there are four isozymes of pyruvate kinase enzyme. There is isoform L, which is present in the liver. There is isoform R, which is present in the erythrocytes. M1 isoform present in muscle and brain, and M2 isoform they are present in early fetal tissue, as well as most adult tissues also express M2 isoforms of pyruvate kinase enzyme. So now let's talk about the hormonal regulation of pyruvate kinase enzyme in the liver. So in the presence of low blood glucose, there is an increase in glucagon level in the blood, which further increases the cyclic AMP level, which activates the protein kinase A enzyme, and protein kinase A enzyme phosphorylate pyruvate kinase enzyme, resulting in the inactivation of pyruvate kinase. So therefore, phosphoenol pyruvate is unable to continue in glycolysis. Therefore, meaning phosphoenol pyruvate cannot be converted into pyruvate, and hence the rate of glycolysis is reduced. As a result, it slows down the use of glucose as a fuel in the liver, but instead phosphoenol pyruvate enters into glycolysis pathway to synthesize glucose, and the glucose which is synthesized through gluconeogenesis is exported to brain and other organs which solely rely on glucose to synthesize ATP. Now on the other hand, in the presence of high blood glucose, there is an increase in insulin level, and insulin levels then increase in insulin secretion activates the protein phosphatase enzyme, which then catalyzes the removal of phosphate group on pyruvate kinase, resulting in the activation of pyruvate kinase enzyme. This leads to the conversion of phosphoenol pyruvate to pyruvate, and it further also result in the increase in the rate of glycolysis. Simultaneously, the gluconeogenesis pathway is inhibited, therefore the inhibited to decrease the synthesis of glucose through gluconeogenesis. So this is the allosteric and hormonal regulation of pyruvate kinase enzyme, which carries out the third irreversible reaction in the glycolysis pathway. So now I just want to quickly review the allosteric and hormonal regulation of glycolysis pathway. So let's first talk about the allosteric regulation. So here we know that hexokinase enzyme is allosterically inhibited by its product glucose 6-phosphate, and it is allosterically activated by its substrate glucose. On the other hand, glucose 6-phosphate is allosterically inhibited by fructose 6-phosphate, and it is activated by glucose, which is similar to hexokinase enzyme. Now let's talk about phosphofructokinase 1 enzyme. So in the presence of when the cell's energy stores are depleted, the accumulation of ADP and AMP activates the PFK1 enzyme, allosterically activate the PFK1 enzyme. And also I forgot to mention that fructose 2,6 bisphosphate, which is also the potent allosteric activator of PFK1 enzyme. Now in the presence of too much of energy, or when cells have abundance of energy, there is an accumulation of ATP and citrate inside the cells. This results in the allosteric inhibition of PFK1 enzyme, hence the rate of glycolysis is reduced. Now, pyruvate kinase is allosterically inhibited by ATP, acetyl-CoA, long chain fatty acyl, and alanine. These are all accumulation of all these molecules are indicative of abundance of energy inside the cells. And pyruvate kinase can also be activated by fructose 1,6 bisphosphate, which is an example of feed-forward reaction. Now let's talk about the hormonal regulation, which is very straightforward, that insulin activates hexokinase and glucokinase enzyme, whereas glucagon level, which increases in the presence of low blood glucose level, it inhibits the hexokinase and glucokinase enzyme. Similarly, insulin activates PFK1 enzyme, whereas glucagon inhibits PFK1 enzyme. And also insulin activates pyruvate kinase enzyme, and glucagon inhibits pyruvate kinase enzyme in the liver. So this is what you need to remember: low blood glucose, high glucagon level. If the glucagon level is high, it reduces the rate of glycolysis. And the main purpose of reducing the glycolysis rate is to stop the glucose consumption by liver and spare that glucose for to be used for other organs, most importantly for brain, because brain solely relies only on glucose to synthesize ATP. Simultaneously, liver can also stimulate the gluconeogenesis pathway to synthesize more glucose, and that glucose again can be used by other organs which only rely on glucose at the time when there is low blood glucose in the body. Now, in the presence of high blood glucose, there is increase in insulin secretion, and increase in insulin level can increase the rate of glycolysis pathway. And the main purpose is to either synthesize ATP through glycolysis, or if there is too much of ATP is being synthesized, then it is being consumed, then glycolysis pathway can also divert into glycogen synthesis in the liver. So then, meaning that the glucose is now being stored as glycogen in the liver, and at the time when there is requirement or when there is low blood glucose, this glycogen can be broken down to synthesize glucose, and then that glucose can be used by other organs. So this is the allosteric and hormonal regulation of three important glycolytic enzymes. Now I just want to quickly talk about the clinical relevance of these enzymes. So we know that red blood cells lack mitochondria, and therefore it only rely on anaerobic glycolysis for ATP production. So red blood cells have this ATP dependent ion pump. So this ion pump uses this ion pumps use this ATP, which is necessary for the maintenance of biconcave and flexible shape of the red blood cells. And because of this biconcave and flexibility of the red blood cells, it is able to travel through the narrow passage of the capillaries. So during hemolytic anemia, there is a deficiency of glycolytic enzyme, and among patients exhibiting the rare genetic defects of glycolytic enzyme, about 95% of patients show a deficiency in pyruvate kinase enzyme, when only 4% of patients exhibit a deficiency in phosphoglucose isomerase. So because of the deficiency of this enzyme, it results in the reduction in the rate of glycolysis, therefore reduces the ATP production, which, because ATP is important to maintain the shape and the flexibility of the cell, that leads to the decrease in the ATP production, leads to the alteration in the red blood cell membrane and changes in the shape of the cell, that causes the phagocytosis of red blood cells by macrophages of the spleen and causes the red blood cells to lyse and leading to the causing hemolytic anemia. So pyruvate kinase deficiency produces mild to severe chronic hemolytic anemia. So when there is a severe form of hemolytic anemia is present, it requires the regular cell transfusion in the patients. So this is something I thought it is very relevant to understand the importance of these enzymes we just talked about. So now the last condition I want to discuss is about arsenic poisoning. So the mechanism of arsenic poisoning involves the interference in the glycolysis pathway, and most importantly the steps where glyceraldehyde 3-phosphate is oxidized into 1,3-bisphosphoglycerate. And remember that for this step it requires the inorganic phosphate. So what arsenic does is arsenic actually competes with this inorganic phosphate, and then by doing that it converts glyceraldehyde 3-phosphate into 1-arseno 3-phosphoglycerate, and the product which is generated here is so unstable that it spontaneously hydrolyzed into 3-phosphoglycerate. So this is the intermediate of glycolysis pathway. So by synthesizing 3-phosphoglycerate, what it does is it actually bypasses the synthesis of 1,3-bisphosphoglycerate. However, remember that glycolysis pathway still continues, but by doing that, it actually loses the ATP molecules which was generated in this step, and therefore the cells which solely rely on glycolysis, they become deprived of energy, and that can have a detrimental effect. So if arsenic poisoning occurs over a brief period of time in patients, they can they have these symptoms which include vomiting, abdominal pain, or diarrhea which can contain blood. Wherever long-term exposure of arsenic poisoning can have more detrimental effect, which can also lead to heart disease or cancer. So this is one of the thing I thought it would be interesting to share, the mechanism of arsenic poisoning, and how arsenic poisoning actually interferes in the glycolysis pathway and make the cells deprived of energy. So thanks so much for watching this video, and I really hope that this lesson was helpful, and if it is, then please like and share the video and subscribe the channel. Thanks so much, and see you soon.